The webinar will begin soon. Please stand by. Good afternoon, everybody. My name is Laura Morell, and I work in the National Center for Emerging and Zoonotic Infectious Diseases at the Centers for Disease Control and Prevention. On behalf of CDC's One Health Office, I'm pleased to welcome you to the monthly Zoonoses and One Health Updates call on February 1st, 2023. Next slide. Although the content of this webinar is directed to veterinarians, physicians, epidemiologists, and related public health professionals in federal, state, and local positions, CDC has no control over who participates. Therefore, please exercise discretion on sensitive content and material, as confidentiality cannot be guaranteed. Today's webinar is being recorded. If you have any objections, you may disconnect now. Links Resources from each presentation are available on our website at cdc.gov/onehealth/zohu/2023/february.html. Next slide. Today's presentations will address one or more of the following 5 objectives: identify an implication for human, animal, and environmental health, identify a One Health approach strategy for prevention of public health threats, identify a One Health approach strategy for detection of public health threats, identify a One Health approach strategy for responding to public health threats, and list 2 ways to improve collaborative practice across the public healthcare team. Next slide. In compliance with continuing education requirements, all presenters must disclose any financial or other associations with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters, as well as any use of unlabeled products or products under investigational use. CDC, our planners, presenters, and their spouses or partners wish to disclose they have no financial interests or other relationships with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters. The Planning Committee reviewed content to ensure there is no bias. The presentations will not include any discussion of the unlabeled use of a product or a product under investigational use. CDC did not accept commercial support for this activity. Next slide. Instructions for receiving free continuing education are available at cdc.gov/onehealth/zohu/continuing education. The course access code is ZOHU Webcast. To receive free CE for today's webcast, complete the evaluation at cdc.gov/TCE online by March 6th, 2023. A captioned video of today's webinar will be posted at cdc.gov/onehealth/2023/february.html within 30 days. To receive free CE for the web on-demand video of today's webinar, complete the evaluation at cdc.gov/TCE online by March 7th, 2025. Next slide. Before we begin today's presentations, Dr. Colin Basler, Deputy Director of the One Health Office, will share some news and updates. You may begin when you're ready.
Thanks, Laura. Good afternoon, everyone. Thank you for joining us for the first ZOHU call of 2023. We appreciate you sharing the ZOHU call website link, cdc.gov/onehealth/ZOHU, with your colleagues from the human, animal, plant, and environmental health sectors, and letting them know about the live webinars, video recordings, and free continuing education this call offers. Before our presentations begin, I'd like to share some updates and resources. You can find links to these resources in today's ZOHU call email newsletter. And if you aren't yet subscribed to the newsletter, please sign up using the link at the top of the main ZOHU call webpage. To begin, CDC's Mpox website details what we know about Mpox in animals and includes information to help prevent virus spread to pets and other animals. You can continue to find on CDC's website the latest COVID-19 guidance and resources for keeping people as well as animals safe and healthy. Next slide, please. Today's newsletter highlights a few One Health focused publications, including a One Health investigation of SARS-CoV-2 in people and animals on multiple mink farms in Utah, and the Lancet series on One Health and global health security. Next slide. Additionally, we've highlighted a recent CDC authored publication in the Journal for American Veterinary Medical Association titled Clinical and Epidemiologic Features of SARS-CoV-2 in Dogs and Cats, compiled through national surveillance in the United States. Next slide, please. We've also shared links to web resources, including the press release marking the end of the Ebola outbreak in Uganda, and a podcast called The Intersection of Humans, Plants, Animals, and Disease with Captain Casey Barton Brevish. Next slide, please. Some upcoming events and observances of interest for February include Responsible Pet Owners Month and Invasive Species Awareness Week, which is happening from February 20th through the 26th. Next slide. Finally, there's our ongoing salmonella outbreaks linked to pet bearded dragons and alfalfa sprouts. There are also ongoing listeria outbreaks linked to deli meats and cheese and enoki mushrooms. Next slide, please. You can visit CDC's Healthy Pets, Healthy People website for a selected list of ongoing and past U.S. outbreaks of zoonotic disease, and for finding details about ongoing U.S. outbreaks on the CDC website linked from today's newsletter. Our next ZOHU call will take place on March 1st, 2023. Please continue to send presenter and topic suggestions for future presentations, as well as news from your organizations to zohucall@cdc.gov. That's ZOHUCALL@cdc.gov. And now I'll turn the call back over to Laura.
Thank you. Next slide, please. You can submit questions at any time using Zoom's Q&A feature. Please include the topic or presenter's name. The Q&A session will follow the final presentation as time allows. You can also email questions to today's presenters. We've included their email addresses on this slide, on the ZOHU Call webpage for today's webinar, and in today's email newsletter. Next slide. Our first presentation, One Health and Wildlife, Current Outbreak of Highly Pathogenic Avian Influenza in North America, is by Dr. Nicole Nemeth. Please begin when you're ready.
Thanks, Laura. Just so everyone knows a little bit where I'm coming from, my background and what I do is as a wildlife pathologist and a wildlife disease scientist. So next slide. It won't surprise you to know that in my mind, One Health, next slide, is kind of focused on wildlife in the center. So really, I consider, wildlife is in close and continual contact with the environment, so we have indirect contact between our domestic animals and humans with the environment, and also even more direct and things that we do as people, and again, with our companion and livestock animals. Next slide. So there are just so many ways that, like I say, as hunters, as occupational, as people using parks and shared areas where wildlife inhabit, there's just so many ways that we contact and have indirect and direct contact. So next slide. So some background on influenza A viruses. And in my mind, this is an amazing example of a One Health pathogen, because as I hopefully will convince you, and if you don't already know, it's kind of at the interface of all these different facets of One Health. And so these viruses are globally distributed, and although you'll find it mentioned that they're in over 100 avian species, it's probably a lot higher number than that. And so there's a broad host range among wild birds, and it really can be transmissible across these bird groups and bird species with no problem. And the historic natural reservoir are birds in the orders Enceriformes and Pteridroformes, which are ducks, geese, gulls, terns, shorebirds. So as you envision, mainly these birds that inhabit aquatic environments. And how it's perpetuated in these natural reservoirs is through fecal or oral transmission, and shared water sources. So a lot of these bird species involved in transmission are either actually swimming and living a lot of their time on these water bodies, or they're right along the edges and again, contacting the water a lot. And you'll hear, HN designations for influenza viruses, and that's these surface proteins that they use, but also important is the pathogenicity that they have. And that is determined by how they cause disease in chickens and not humans and wild birds, which are really important in transmission and as we'll talk about their health impacts. And interestingly, we don't talk about it a lot, but there are some ongoing studies to show that wild birds do have immunity to some of these different high path and low path viruses, and that may help them and alter transmission dynamics. Next slide. And so the environmental component for these viruses is very important, which we've already kind of talked about a little bit. So these infected birds are shedding the virus through really oral secretions and feces all the time, as you can imagine. I have birds at home and they poop all the time. So they persist in water, these viruses, and that includes really cold and frozen water. And the viruses can withstand, certain levels of conditions that have been well studied, like salinity and pH, and again, temperature. So they can even persist for over a year. But really all these viruses need are shorter term persistence, which allows the virus to kind of be maintained among birds passing through, shared areas, shared habitats, again, often involving aquatic environments. So that is one factor that really allows this virus to continue and wreak havoc. And so you don't need direct bird-to-bird contact for the maintenance of transmission because, again, it's in the environment. And I have been asked in the past, especially now with the ongoing outbreak, how long does it maybe survive in a carcass? And we really don't know the specific answer to that, but really it survives as long as it may take for the carcass to break down a bit and be consumed by something else that may get it via that route. We'll talk about that shortly. Next slide. And so the current strain and the current outbreak that we're experiencing in North America is with the strain that was listed here at the top, highly pathogenic goose guangdong virus that was first detected in 1996 in Asia. And so I don't want to go through specifics of the history there, but there have been some expansions, and even in about 2005, the virus was detected in Western Europe and the Middle East and Africa. So it has shown ability to spread long distances. And the bottom line is it's had quite a number of years now to evolve, and it's continually evolving and reassorting as we speak. And we did have an outbreak here in the US and affecting Canada starting in November 2014. And that was the most noteworthy outbreak probably in most of our minds. And it really caused a huge economic loss to the poultry industry. And during this outbreak, there were limited reports in wild birds. There were some, but weirdly, as there's a publication on the right that just kind of goes into this a bit, you can see the blue states are the ones where there were wild birds or poultry cases, as well as again in Canada. The virus seemed to kind of disappear after about 2015. And no one really knows, again, how it came or why it disappeared. There were a lot of efforts in the poultry industry to stamp it out, and that was successful. But again, in the wild birds, no one really knows. It kind of came and went, so to speak. And all was quiet. People breathed a very big sigh of relief until a little bit over a year ago, when again in Canada, this time on the eastern side, there was a detection of this virus killing birds. And then rapidly spread southward and westward. So it didn't take too long before it was in Florida, and I'm here in Georgia, and that's when we started working on it. So this outbreak is ongoing. Again, we didn't know with the 2021 event and ongoing since then what would happen, and people were waiting, and what we've seen is that it is not going away easily this time. Next slide. And so I wanted to bring to your attention that USDA has some excellent information on their website. And if you Googled USDA highly pathogenic avian influenza, you would come to these maps and some data, and you'd get a lot of information. And it's updated regularly. Canada has something very similar, and it's been detected in all Canadian provinces. But I just want to mention the caveats to the USDA website and data are that some of the detections in there are from healthy hunter harvested waterfowl, quite a number of them actually. So it's a mix of detections of both birds that have died and other birds that have been survived and just were healthy and either hunted or maybe swabbed and released. And so some of these detections, though, I want you to think the single detections on this map also may represent hundreds to thousands of wild birds that have died. We have tested some where like one snow goose came in, it represented observation of what was estimated to be thousands of birds in the field. And the website lags a little bit only because they have to confirm every case, and there's a lot of cases. Next slide. So just to point out that the majority of these detections are waterfowl. Again, some healthy birds detections, but a lot of them fatal infections. And a lot of the ones that I know of that are fatal are like snow geese, Ross's geese, lesser scaup, Canada geese. Then raptors is the next most largest group, and that's been a lot of eagles, a lot of black vultures, but many species of pretty much all raptor species you can think of, including ospreys and screech owls and everything else. Next slide, please. So also to point out, I don't do this enough in this talk, but I do want to mention poultry, because this influenza viruses are often devastating to the poultry industry, and this outbreak is no exception, especially highly pathogenic strains. And as of a couple weeks ago, there were almost 60 million birds affected. And this is domestic birds, poultry, commercial flocks, and backyard flocks affecting most states and ongoing. So they are doing intensive efforts as they do to get eradicated in these premises and quarantine and again cull. So that's where we stand right now, is nearing 60 million birds have to be culled. Next slide. And I also would just want to bring to your attention that this virus has been detected in some wild mammal species, interestingly, and not surprising though, because most of the species are carnivorous or mesocarnivores that are probably just kind of incidentally getting it through consuming infected waterfowl or other birds possibly. And there haven't been that many, so it's probably not a very common event, but some of them have been fatal and encephalitis, severe lesions, and neurologic disease. And about half of these have been red foxes, but there's been a variety, including, even believe it or not, a bottlenose dolphin down in Florida. So there's that water component playing a role likely. Next slide. So getting to the human aspect, this virus is considered by CDC to pose a very low threat to human health at this point. It's been very rarely documented in humans, and including humans that are diseased, but there is a range of manifestation from asymptomatic to fatal. And it's going to enter kind of similar to the birds, although the birds we think fecal oral most likely and ingested, but for us it could enter via eyes, nose, mouth, so really any mucous membrane, or be inhaled. And that's generally going to occur when there's unprotected exposure. So there are things we can do to protect ourselves and to advise others, which we'll talk about. But most of the human cases, there have been a few more in Asia, and these were back earlier in outbreak periods of 1996, 2005 during the spread, and generally people that were in very close contact with domestic fowl, to the point of like holding them, and having them close to their face and things like that. As far as the current outbreak timing, there has been a case in the UK, which was actually someone that had raised birds that were infected. That was an asymptomatic infection. And more recently in the US, we've had one human case, and that was someone that was involved in culling infected poultry, so very close contact and probably handling a lot of them, had fatigue and then recovered, luckily. Next slide. So the recommendations follow exactly what you'd think from what we just said, is that if you can wear some personal protective equipment, what we do for necropsying a lot of wild birds that come in is, again, face shield, gloves, mask, KN95 if you have it, or N95 is better, obviously, but any kind of mask, gloves, and then wash hands with soap and water after handling. Don't touch your face. It's very similar to SARS coronavirus in a way. And then really try to be very conscious about your clothes and anything that might be on you if you are even the chance of having any contact with any domestic or wild birds at that point. Next slide. And then getting to the wild birds finally, which I already confessed was my favorite aspect. Vast diversity of affected species. It is a neurotropic virus, so it goes quickly to the brain, and so these birds do exhibit neurologic disease. Predatory and scavenging birds have an additional transmission risk, which we'll talk about a little bit more, through consuming dead infected birds. But the disease is multisystemic, so we detect it in pretty much every tissue that we look for it in if they die of it, and it really, they can die so quickly that we've had reports of birds falling off the trees, and they usually have secondary trauma because they've just hit the ground. And we detect it against swabs. Oral colloquial swabs are the standard for birds, but sometimes we get it high, like a lower CT value in the brain, so a higher kind of virus amount, presumably. Next slide. And these are the videos, which they should click on, like, do a next, and then do a next slide again. I think it'll play them. There we go, concurrently. It just kind of is to give you a little bit idea. That one's a little bit slowed down, but that poor bald eagle's head is just kind of continually shaking. And the duck as well, like he's just having some head tilt, head tremors. And so this is what people are seeing. They're weak and they're lethargic. Sometimes they're seizing. So well, poor duck finish. Yeah, thank you very much. Next slide. And so that's what people are seeing. So we'll talk really briefly about what that has entailed with kind of human wildlife interactions. But through these detections and this high mortality in eagles, I just wanted to point out, because this is kind of a special project that we've looked at, is that we noticed last year that a lot of the eagles that were nesting were dying, their chicks were dying, there were a lot of nest failures, and it made us quite nervous that we need to keep an eye on eagle populations in this regard, because the eagles obviously spend a lot of time around water, even coastal areas where some of these waterfowl are in high congregations. So they may be a sensitive ecological indicator species as well, because they were one of the earliest species in the US that we saw with documented high path AI. And again, think about it that they are very opportunistic scavenging birds. They're smart, so they go for the easy stuff. And so they were eating a lot of dead and dying waterfowl for sure. So we are keeping an eye on potential, we need to keep an eye on potential long-term effects. Next slide. And so this slide is just kind of to show you that this has been a sad thing to observe and distressing to people. And in Florida, for example, there's a lot of volunteers that monitor eagle nests. So the top right A is an attentive pair of eagles watching their chick off in the corner there, and he's healthy. The next day, he was found dead under the nest, tested positive for high path. 5 days later, the adult female from that nest was again found dead under the nest, which is there's a wildlife officer retrieving that carcass. And then the lower right is just a eagle in a rehabilitation that did test positive for high path, did die the next day after that, that picture. Next slide. And then the last kind of interesting scenario I wanted to show you is these black vultures, which more in the eastern US, but black vultures have been dying in the hundreds and thousands probably. And something interesting happened with them, because they were probably consuming dead waterfowl at the beginning, but they're in these roosts with hundreds of birds. And once the vultures themselves started getting it from the waterfowl consumption, they started dying and consuming each other, because they're not very discriminatory in that regard. And it just maintained this cycle that perpetuated without really any outside influence or force or, virus introduction into these roosts or these groups of vultures. It was just simply, they would stay in the roost, and a lot of these roosts are near landfills. So there was, a lot of humans watching this happen. So this was an interesting thing. We're keeping an eye on this as well. And Dr. Mark Cunningham at Florida Wildlife Conservation Commission is doing a lot of work on this. So just again, implications of vulture impact, population impacts, because that may be occurring, and they generally clean up a lot of stuff for us. And so without them, it might not be a pretty picture. Next slide. So finally, I'm taking this picture, if you will, because this is an area where these dead vultures are dying along the shore of a river in Florida, and these people are just kind of kayaking along right next to it. And next slide. And I just wanted to mention Florida Wildlife Commission's efforts, and they had tried to remove carcasses. It became logistically unfeasible because there were so many. They also educated people, put a lot of messaging out, limit access to these affected areas. They cordoned off areas, as you can see in the top picture. They warned people not to handle live or dead birds. The human aspect was difficult there, because people wanted to help these sick birds. So they wanted to pick them up. They were bringing them into their houses. So that was tough, and they had to say, please let nature take its course, so to speak. And they asked rehabilitators again to euthanize upon arrival any suspects. So they came to the conclusion that really information sharing is key. The lower picture is an incinerator right on site trying to keep up with the carcasses, but just suffice to say, so many efforts have gone into trying to figure out what we can do about managing it and the environment, and that is very difficult, if not impossible. Next slide. And just to wrap, just to thank some folks here at the Southeastern Cooperative Wildlife Disease Studies, specifically Dave Stelnick and Becky Pelson, and Mark Cunningham and Bob Sargent with their respective wildlife agencies. And thank you guys for going on that whirlwind. Appreciate it very much, your attention.
Thank you. Next slide, please. Our next presentation, Environmental Fungicides and the Development of Antifungal Drug-Resistant Mold Infections in Humans and Animals, is by Dr. Jeremy Gold. You can begin when you're ready.
Thanks so much. So good afternoon. As you just heard, my name is Jeremy Gold. I'm a medical officer with the Mycotic Diseases branch at CDC. And today I'll be talking about the link between fungicides in the environment and the development of antifungal drug-resistant mold infections in humans and animals. Next slide, please. So I'll start with some background about Aspergillus, as I know everybody might not be familiar. Aspergillus is the most common type of severe mold infections in humans. Aspergillus is caused by the environmental fungus Aspergillus fumigatus. This disease is uncommon, but can be life-threatening. It usually involves the lungs and mainly affects people with severe immunocompromise, like patients who've had a stem cell transplant or a hematologic malignancy. Next slide. Notably, aspergillosis is increasingly identified as a cause of illness and death in other types of patients, including those who are critically ill with the flu or COVID-19. Next slide. Aspergillus also causes disease in animals. It can cause bronchopneumonia, sinonasal aspergillosis, invasive pulmonary aspergillosis, and otitis in various animal species, and it's a common cause of death among penguins. Next slide. Fortunately, drugs that are called triazole antifungals exist, and triazole antifungal drugs include voriconazole, posaconazole, isavuconazole, and itraconazole, and these are the first-line treatment for invasive aspergillosis and the only orally available therapeutic options. The primary alternative drug, called amphotericin, has major side effects, particularly causing severe kidney damage. And triazole drugs are critical for treating aspergillosis and saving lives, drastically improving patient survival. Next slide. Unfortunately, triazole resistance is an emerging problem associated with decreased patient survival. The data in this slide are from a 5-year retrospective cohort study in the Netherlands, and they showed that patients with azole-resistant aspergillus infections had a 33% lower survival rate compared with patients who had azole-susceptible infections. Next slide, please. Fungicides are used in agriculture and numerous other settings. And those settings include horticulture, personal gardening, and also wood and paint preservation, just to name a few. Fungicides in agriculture serve a very important role in protecting crop health and the food supply. However, mounting evidence has implicated the use of triazole fungicides, which are chemically similar to antifungal drugs used in human and animal medicine, as a cause of emerging triazole drug resistance. And something that's interesting is that Aspergillus fumigatus is not itself a plant pathogen, but it's found throughout the environment, especially associated with plant debris. And the Aspergillus fumigatus can develop resistance to azole drugs that are used to treat the same azole drugs that are used to treat aspergillosis in humans and animals when the fungus is exposed to azole fungicides sprayed on crops. Next slide. So Aspergillus fumigatus strains that develop resistance through exposure to azole fungicides harbor unique mutations in the target gene CYP51A. And the two most widely dispersed CYP51A mutations are the TR34, that stands for 34 base pair tandem repeat in the promoter sequence, and TR46, which represents a 46 base pair tandem repeat in the promoter sequence. And these mutations in the Aspergillus DNA confer resistance to itraconazole and voriconazole, two of the main triazole antifungal drugs, respectively. But are some of these, there are also other mutations that can confer pan-triazole resistance as well. Next slide. So what happens is patients, including those who've never taken a triazole antifungal drug themselves, the patients can breathe in Aspergillus fumigatus spores that are already triazole resistant, breathe those in from the environment, and develop severe infections. Next slide. For the sake of time, I wasn't planning to go through all the evidence supporting the link between environmental fungicide use and the emergence of triazole-resistant antifungal infections in humans and animals, but it's very well summarized in a report from the European CDC. It's called the European CDC 2013 Risk Assessment, and that's available for free online and kind of outlines all the nitty-gritty evidence. Next slide. Of note, in the United States, the use of triazole fungicides has increased fourfold from 2006 to 2016. And there are several factors that might explain this dramatic increase in US triazole fungicide use, including increased corn production in response to higher prices, plant diseases in certain regions, the ability to use new fungicides on field crops, and in particular, marketing of fungicides for use on field crops. Next slide. Strains of Aspergillus fumigatus with environmentally linked resistance mutations have been detected in the environment in countries across the globe. Notably, the Netherlands was actually the earliest hotspot for this problem, and that's thought to be related to the high use of triazole fungicides related to the country's thriving tulip industry. But as you can see here, these resistant Aspergillus fumigatus with the environmentally associated mutations, those have been found kind of throughout the globe. Next slide, please. In the United States, surveillance for triazole-resistant infections is currently still in development, and we are lacking data on the prevalence of human and animal infections. There have been several human infections detected in the United States, recently including one fatal infection in a patient in Pennsylvania. And the data from these reports, though, come from passive surveillance, and the true scope of the problem in the US is yet to be defined. Next slide, please. Triazole resistance has been detected in animals too, although the studies are even more limited. These articles highlight the detection of azole-resistant A. fumigatus from a bottlenose dolphin. I feel like we heard about a bottlenose dolphin in the last presentation too, so maybe that's like a theme today. But anyway, they've detected these mutations or these resistant organisms from dolphins and geese, actually in Poland. Next slide, please. So the problem of azole-resistant Aspergillus fumigatus is gaining increased attention. For example, it was added onto CDC's 2019 antimicrobial resistance threats report. I just put that here to highlight that this is really an issue that's garnering national and international concern. Next slide. And currently at CDC, we're working to increase surveillance for resistant infections through the Antimicrobial Resistant Lab Network, or the AR Lab Network, as it's sometimes known. And since June 2021, clinical laboratories across the US can submit Aspergillus fumigatus isolates to 2 regional laboratories for susceptibility testing. And more information about how to submit isolates, Aspergillus fumigatus isolates to the AR Lab Network can be found on the CDC website, or just by Googling AR Lab Network Aspergillus. Next slide, please. We're also hoping in the near future to expand surveillance for invasive mold infections in the United States. We're looking to support studies that assess the prevalence of azole-resistant infections in US patients, and conducting surveillance through the Emerging Infections Program, which is an active surveillance network. Currently, the Emerging Infections Program, or EIP, surveillance for mold infections is just limited to the metropolitan Atlanta area. We're hoping to expand this program soon as well. Next slide. And we've also been supporting efforts to perform environmental sampling for azole-resistant Aspergillus fumigatus in the US. For example, CDC funded some University of Georgia researchers to collect samples in the southeastern US, and it was interesting. They found azole-resistant Aspergillus fumigatus in peanut fields. But we're hoping that we can see further studies with broader sampling strategies to get a better sense of the environmental distribution and prevalence of azole-resistant Aspergillus fumigatus in the United States, and also to more clearly to cement the association between human and animal infections and the prevalence of these organisms in the environment. Next slide, please. So to kind of sum it up, antifungal drugs, including the azoles, are critical in are critical to the health of humans, animals, and plants to reduce the burden of disease. And we're in the earlier stages of really addressing this issue in the United States. It's going to take a One Health approach, a cross-sector approach, both to improve the stewardship of triazole drugs in human, animal, and agricultural practice, and also consider surveillance efforts need to also be performed through using a One Health lens too. Next slide. And that's all I have for today. Thank you so much for listening. I really appreciate your attention. And please do feel free to reach out or shoot me an email if you'd like to discuss this further.
Thank you so much, Dr. Gold. Next slide, please. Our final presentation, Knowledge, Attitudes, and Practices of SARS-CoV-2 in Animals Among Veterinarians in Los Angeles County, is by Allison Joyce. Please begin when you're ready.
Thanks, Laura. Next slide, please. In 2022, the LA County Department of Public Health Veterinary Public Health Program received funding from the CDC and the Council of State and Territorial Epidemiologists to conduct SARS-CoV-2, or the virus that causes COVID-19, surveillance in animals. A large part of this funding went towards testing various animal species in LA County for SARS-CoV-2, hoping to answer interesting questions like if SARS-CoV-2 is circulating in any animal species in LA County, and if so, are they infected with the same strains seen in humans, or are there mutations? And as part of this grant, we also conducted a SARS-CoV-2 in animals knowledge, attitudes, and practices survey among veterinarians in LA County. We really wanted to see what local veterinarians knew about SARS-CoV-2 in animals, what their attitudes were about it, and specifically regarding testing animals for SARS-CoV-2, and what they were actually seeing and doing in practice. Next slide, please. And before I get into the results of the survey, I wanted to share some information about our response rate. So starting in June of 2022, we invited veterinarians from the over 500 known vet clinics, animal shelters, and animal rehabilitation centers to participate in this survey. Over the course of 5 months, we tried many different avenues of promoting the survey. We sent personalized emails, faxes, notifications through our Animal Health Alert Network. We received assistance from the Southern California Veterinary Medical Association in distributing the survey. And we actually started including a blurb and eventually an infographic promoting the survey at the bottom of emails when we alerted vet clinics of rabid bats in their area. In the end, we received 152 complete responses, which gave us a response rate of 26%. And the map on the right depicts our response rate, where each boundary represents a zip code in LA County. The gray areas represent zip codes in LA County with no known veterinary practices, and bright red represents areas where none of the known veterinary practices within that zip code responded to the survey. And the shades get lighter all the way to an off-white, which represents areas where 76 to 100% of known vet clinics in that zip code responded. I do think we got a fair distribution of responses across LA County, although there are a few pockets of concentrated red. There are some larger zip codes up towards the northern region where we did not reach very well, and there's also a pocket of concentrated red in the southeast. Next slide, please. And now I'm going to get into the results of the survey, starting with the knowledge section. Next slide, please. First, we provided a list of animals, which can be seen across the bottom, and asked veterinarians to identify which have been found to be naturally and/or experimentally infected with SARS-CoV-2. Their responses are shown on this bar graph, with navy representing natural infection and orange representing experimental infection. And I've also included the correct answer for each species, which can be shown with an appropriately colored circle next to the animal name on the x-axis. With the exception of chickens, every animal listed here has been shown to be either naturally or experimentally infected or both with SARS-CoV-2. Over half of our responding veterinarians knew that big cats, dogs, and domestic cats have been found to have natural infection with SARS-CoV-2. And 20 to 30% also identified ferrets, gorillas, mink, and white-tailed deer. Other species shown to be naturally infected, including otters and hamsters, as well as the species shown to be experimentally infected, were generally not known. Also, 36% of veterinarians identified bats as capable of natural infection, which actually hasn't been proven yet. Egyptian fruit bats have been experimentally infected with SARS-CoV-2, but there is currently no published evidence of a natural infection in a bat. Next slide, please. We asked veterinarians to identify common signs of SARS-CoV-2 in animals. The majority of veterinarians correctly identified cough, fever, sneezing, lethargy, and nasal or ocular discharge as common symptoms. A little less than half identified inappetence, and 30% indicated shortness of breath as a common symptom. Next slide, please. We also provided a list of statements about SARS-CoV-2 in animals and asked veterinarians to select whether the statement was true or false. When asked, animals can be asymptomatic and still be infected with SARS-CoV-2. Over 80% of responding veterinarians knew this was true. One of the most interesting results we received from our survey was the response to this statement. In multiple surveys of pets in COVID-19 positive households worldwide, up to 40% of dogs and cats are reported to test positive by serology. This is a true statement, but only 15% of responding veterinarians indicated it was true. 42% thought it was false, and 43% indicated they did not know. Next slide, please. And now I'll get into the results from the attitude and practice sections. Next slide, please. When asked if an animal tested positive for SARS-CoV-2, would that change their course of treatment? Less than 20% of veterinarians said yes, usually indicated that they would recommend isolation. 60% said maybe, and 20% said no. Veterinarians indicated their course of treatment would not change because the standard treatment for a viral infection is supportive care. Therefore, knowing the viral agent causing the disease would not really change their treatment. And when asked if they thought SARS-CoV-2 testing was useful in animals, over 60% of responding veterinarians indicated yes. Next slide, please. We provided veterinarians with a scenario. An animal comes in with respiratory signs and asked if they would include SARS-CoV-2 in their differential list. 10% indicated they would for all animals that come in with respiratory signs. 55% indicated they would if the animal had a known exposure to SARS-CoV-2. And 35% indicated they would not include SARS-CoV-2 infection in their differential list. Next slide, please. We provided a list of scenarios and asked veterinarians to indicate which would make them suspicious an animal was infected with SARS-CoV-2. 68% indicated if the animal has respiratory symptoms, and they've ruled out other diseases. And 90% indicated if the animal was exposed to a person with current or recent COVID-19. When asked if they asked their clients if anyone in the household recently had COVID-19, 25% of veterinarians indicated they do ask about exposure for all animals that come in. And this increased to 42% when the animal comes in with respiratory signs. Next slide, please. When asked if they thought it was challenging to test an animal for SARS-CoV-2, 33% indicated they did not find it difficult, and 35% indicated it was difficult because owners find the test expensive. Results from our survey found that the average cost of a SARS-CoV-2 test for a pet was $307, with a range from $150 to $500. When asked what barriers they have actually encountered to testing animals for SARS-CoV-2, 25% said the owner declined due to cost, but almost 2/3 said that they had not encountered any animals that they suspected of having a SARS-CoV-2 infection. And in fact, only 14% of responding veterinarians indicated their practice had seen an animal they suspected of having SARS-CoV-2 infection. Next slide, please. And now I'd like to discuss some of the key findings from this survey. Next slide, please. One of the key takeaways from our survey was that most veterinarians in LA County were not aware of the many different animals that are susceptible to SARS-CoV-2 infection, or that humans infecting their pets can actually be quite common. The CDC, USDA, and OIE all have excellent information on SARS-CoV-2 in animals available on their website. And I suspect many organizations, including local health departments and veterinary medical associations, perform education and outreach via email and social media accounts. Many researchers across the world have conducted studies on animal susceptibility to infection with SARS-CoV-2, and this data is available in peer-reviewed literature. Our survey suggests these methods may not be effectively reaching practicing veterinarians, especially during a time when veterinarians are swamped. Sending an email, updating a website, or publishing a paper may not be reaching our One Health partners as well as we might think. The main barrier for testing animals for SARS-CoV-2 identified in this survey wasn't a barrier to testing itself, but that veterinarians were not seeing animals they suspected of being infected with SARS-CoV-2. This is likely due to a myriad of reasons and cannot be fully teased out from the data from this survey. Our survey did find that most veterinarians are not aware how common SARS-CoV-2 infection can be in companion animals. 1/3 of vets don't include SARS-CoV-2 in their differential list for animals that come in with respiratory signs, and more than half don't ask about exposure to a person with COVID-19. But there are likely underlying factors at play that were not captured in this survey. For example, the isolation people keep when they test positive for SARS-CoV-2, as well as the mild or even asymptomatic presentation in animals, likely contribute to pets not being taken to the vet for what could be a SARS-CoV-2 infection. Also, early messaging to veterinarians from public health organizations did not recommend routine testing of pets for SARS-CoV-2. This was partly based on shortages in PPE and testing agents, but also because we weren't really seeing disease in animals, despite millions of cases of COVID-19 in people worldwide. And we didn't have these studies showing that pets do catch SARS-CoV-2 from their owners. It just usually presents asymptomatically. I think it's been challenging to shift away from that message of not testing animals once it was set, especially as COVID fatigue and burnout set in. However, our study also found that 60% of veterinarians thought that SARS-CoV-2 testing in animals was useful, and this was despite it not likely affecting their course of treatment. This is good news. Even if the current risk of an animal transmitting SARS-CoV-2 to a human is low, and if the disease in animals is likely to be asymptomatic or mild, it is important from a public health standpoint to conduct surveillance for disease in animals, especially for an emerging zoonotic virus like SARS-CoV-2 that has a well-established ability to infect multiple different species of animals, as well as mutate and form new subvariants. Engaging with veterinarians is a key part to conducting this surveillance. But if we want to utilize these relationships, we may need to identify more effective ways of communicating with practicing veterinarians and consider implementing methods to increase reporting either through mandates or partnerships with diagnostic laboratories. And thank you all very much for your time.
Thank you very much for your presentation, and thanks to all of today's speakers for your informative presentations. Next slide, please. Links to resources from each presentation are available on our website at cdc.gov/onehealth/zohu/2023/february.html. We do have time for questions. Just a reminder to please use the Q&A feature in Zoom to send your questions and include the presenter's name or topic. We'll start with a few questions for Dr. Nemeth. We had a couple questions about environmental conditions favoring persistence of the influenza virus. Can you talk a little bit more about which environmental conditions do favor persistence?
Yeah, so good question. There have been quite a few laboratory studies published on that with controlled setting with the water, and then a more recent one in Alaska with some wetlands. And it boils down to, it seems like temperature is probably the most important effect on the persistence, but there are when you start looking at multiple variables, there are interactive effects between like temperature and salinity, and even pH, and also organic material in the water. So a lot of these studies, they find that it persists better in the surface water, versus sinking down where there's more organic matter. And there's like, for example, cold water persists better than warm higher salinity water. It doesn't persist as well. Cold, higher salinity water persists a little bit better. So again, you kind of have multiple conditions, obviously, in the water, but bottom line, I think, a lot of it's temperature, with colder temperatures are better for the virus.
Thank you. So given that, are there any concerns for the spring and summer for people who may recreate in lakes or around areas where there have been large scale die-offs that occurred in the winter or early spring?
Yeah, that's a really good question. And one thing I would suggest is, in order to know, if there are where these birds are dying off and if it's due to high path, I would probably, if you're going to be out more remote or in aquatic environments, you could contact your state wildlife agency possibly to get that information. And I mean, on a practical standpoint, I know this kind of goes without saying, but if I was in an area where I saw multiple birds, dead and dying, I probably myself would avoid, contact with the water where, you're submerging yourself and it's contacting your mucous membranes. But in general, I will tell you that even influenza experts I've talked to have said pretty emphatically that water doesn't as much, the environmental kind of exposure route doesn't pose so much as a risk to humans as it is more a risk to both poultry or wild birds that may be in contact with the water, and that may be a matter of kind of duration and extent of contact. So again, on the practical side, I probably would avoid, if you knew there were multiple birds dying in an area, maybe not go in that water as a general recommendation, but it's still considered low risk as far as human risk factor of infection via water.
Thank you. Okay, switching gears to Dr. Gold, we have a few questions for you. Are most of the triazole resistant infections in humans thought to occur via outdoor exposure?
Yeah, that's a great question. I think the honest answer is that we don't exactly know, and we need to do a lot. We are hoping to support a lot more sampling in various environments, be they indoor or outdoor, to determine kind of where it is that azole-resistant molds are most likely to live. Most of the work and exposures that have been documented have been in the outdoor environment, and I think that's because most of the largest kind of volume of use of antifungal or of fungicides is in the outdoor environment. So that's where we think people are most likely to become infected or to be exposed to resistant molds. But the tricky thing is these molds, the Aspergillus fumigatus spores, can travel for miles and miles. So it can be hard to tell from where the patient was actually exposed to the pathogen. So we don't know, but probably outdoor, but definitely needs more research is the take home.
Thank you. And is there any tracking of antifungal use in regions and environmental sampling to ID resistant organisms?
Thanks for that question. So I guess there's a couple of pieces in there. One source of data that we look at least track the use of fungicides. Well, if we're talking about use of antifungals in agriculture, for example, we look at the U.S. Geologic Survey, which is where we were able to derive our estimates for agricultural pesticide use. And we also look at other data sets like IQVIA and sort of other commercial insurance claims based data sets to look at antifungal drug use in humans. And we recently got access to a data set that looks at sort of, I believe we've been trying to look at veterinary use of antifungals as well. We are really hoping a major area of interest for our group at CDC is to sort of help either support or perform more environmental sampling to see where the azole-resistant strains are living in the environment. So we have a couple sort of different mechanisms through which we're trying to promote that kind of surveillance and research, but not a lot like actively going on. There's some academic groups that work on it that we collaborate with who are wonderful, but it's an area that we're very interested in.
Thank you. And then we have a question for Allison as well. Did you break down demographics or career focus of the veterinarians that were surveyed? For example, small animal focus versus public health versus mixed practice, or shelter versus non-shelter vets?
Yes, thank you. It's a good question. We kept our survey anonymous, so we didn't collect too many demographics, but we do have the zip code from each veterinary practice that responded. And we did include a question asking what area of veterinary medicine they currently practice, giving several different options, including like small animal, zoo, large animal exotics, and that kind of thing. And most of the responses were from small animal practices. And then we also had a few from shelters.
Thank you. And going back to Dr. Nemeth, we had a couple questions about vaccination. Is there a vaccine in development? Is there any research on avian vaccination of susceptible endangered species?
Yeah, really good questions. And I have to admit, the poultry side of the vaccinations and influenza is not my area that I wish I could give you more information, but I do think there are some vaccines in poultry. As far as I know, some of them aren't used in the US or any of them as much because of the surveillance aspect, because it would interfere with surveillance. I think there are some countries in the world that use. So my point being that there are some, that have been developed for poultry, since it is such a huge disease in poultry and pathogen of risk. As far as endangered species and worrying about maybe even like captive zoological collections or birds, yes, in the wild that are of high value or at risk of threatened endangered status. I can mostly speak from past experience with West Nile virus. It is very challenging, and what is developed is usually, more for the domestic species. So it's kind of considered off label for wildlife, and people do start testing those sometimes when these viruses become a risk. I haven't heard of that happening yet for this current high path strain, at least in North America. I think it would take a lot for people to go that route, but I do think you're correct that, it's logical that it come to mind in trying to protect birds. And it's just so difficult to try to, protect wild birds from these things and even feasibly vaccinate. And what happens with these waterfowl and these wild birds too is they're continuing adding naive birds to the population every year, too, and especially when they're passing through these migratory sites, they're just always adding these naive birds into the population, which again makes vaccination very difficult. But so it's a good question. I haven't heard specifically about development for use in non-domestic birds. But sometimes different research groups trying to develop, different strategies for how to do that. So I wouldn't be super surprised if that didn't start happening.
Thank you. And then we've also had some questions about how this might affect conservation activities for endangered or threatened birds. Can you speak to that a little bit?
Yeah, so as far as, worrying about different wild bird species and their conservation status, it kind of goes back a little bit to what we talked about. Is at least right now, what we can see is that the major groups are waterfowl, some aquatic birds, for example, in the category of like herons and egrets and pelicans that have also been infected, but at much lesser numbers. So it'd be more waterfowl that might be endangered that you'd worry about, or the raptors. I do think it's a concern for some raptors, and it's always going to be thinking about the biology and the ecology of the species as far as their risk level. In terms of, especially, it's probably most likely those that are either frequenting, aquatic environments a lot and hanging out there, or those that are consuming, infectious waterfowl. So, I think that needs to be considered, but it's just such a wide open story right now where we just need so much more information collected, again, with collaborations with folks in the field and biologists and wildlife managers and natural resource folks, and just to try to collectively get a better idea about the risk. And it's going to take time, because this year that we've just experienced, none of us knew at any point if it was just going to disappear again, if it was going to come around again in the fall or the spring. And right now it is. So it's kind of the risk we can't really determine right now, because we need more time, which is also causes panic, of course, among people that want to conserve wildlife. So I understand that, but there's so much left to be known. And again, that we did that small initial project in bald eagles basically to give a warning, like we need to pay attention to this. So I think just kind of trying to be aware of it from a conservation standpoint, but not panic yet. Like we don't have strong indicators yet at this point that it's a concern, but we need to really keep a close watch.
Thank you. And then we have time for one last question for Dr. Gold. Are there any available tests for resistant A. fumigatus that can be used in public health labs or critical care units?
Sure, thanks for asking that. So there's a few different ways that you can do testing for azars, or really just two ways. But in terms of what's available in public health labs and clinical settings, unfortunately, the capacity for most clinical laboratories don't have capacity to do susceptibility testing for A. fumigatus and other molds. However, it is available for free for any like free of cost for any clinical specimens where you have an Aspergillus fumigatus isolate. They can be sent to the AR lab network. And currently there are two, they can be sent from anywhere in the country. There are two members of the lab network that are actually doing the testing, Tennessee and Maryland, and they can be sent there. And what they'll do is they'll screen they'll screen the isolate for resistance, and those isolates that test, that screen positive, they undergo broth microdilution, and to determine minimum inhibitory concentrations. And then if there's resistance, then they undergo genetic or molecular testing to look for like specific mutations. So that testing is available. We're hoping to build up the capacity across the country more so. But trying to speak fast so we don't run out of
Thank you.
You know me if you have questions, happy to talk about it.
Thank you. And that's all the time we have for questions today. But if you have other questions for our presenters, we have included their email addresses on this slide. They're also on the ZOHU call webpage for today's webinar and in today's email newsletter. A video of today's webinar will also be posted within 30 days. Next slide, please. Thank you for joining our call today. Please join us for the next ZOHU call on March 1st. And we, again, thank you for your participation. This ends today's webinar.